Thank you, Eva. We have our second speaker. Dr. Chris Bahl is a protein designer, a biochemist, and a structural biologist, and he's pioneered the ability to computationally design synthetic mini proteins de novo. He was trained at the Baker Lab, and he's formerly the head of protein design at the Institute of Protein Innovation at Harvard Medical School. And today, Chris is the chief scientific officer and co-founder of AI Proteins, which leverages unique and powerful synthetic mini protein modality to rapidly create novel therapeutics. And with that, a round of virtual applause for Chris.
Thank you so much, Sally, for that kind introduction and for the invitation, the organizers, the invitation to share our work today. So, share here. Yes, so I was an academic until last fall and decided to make the leap to biotech, and was very fortunate that the members of my lab essentially all came with me on this journey. So I'm gonna share some work today. Story is a bit foundational on creating mini proteins and the ability to design them, and then I'll share some really raw cutting edge data that's just come out of our work here at AI Proteins, just to try to make this a little bit interesting. So the talk for today, first I'll introduce mini proteins. This may be something that maybe not everyone in the audience is familiar with. And then I'll review the ways that we can design mini proteins de novo. And then I'll talk about the next gen mini proteins that we're creating here at AI Proteins. Okay. So to kick things off, I find a good way to think about mini proteins and start getting your mind in the right place is to think about them in context of other molecules that may be more familiar. And I know this is a peptide audience, so this is probably a space that many of you are thinking about already. So I'll be very brief here. So on the small side, we've got small molecules as kind of the prototypical drug here. We've got aspirin representing this very large and diverse category of therapeutics. But there are a couple of features that, well, not absolute, but tend to be quite unifying throughout the small molecule space, right? So generally we're targeting enzymes or receptors with small molecules, usually pretty cheap to manufacture, quite stable. You can administer them orally. And there's the potential for some side effects, right? Getting really specific binding with small molecules can be challenging sometimes. And then you also have to worry about the small molecule being processed into something that has toxicity. On the other end of the size spectrum here, we've got large molecules, and representing large molecules broadly here are antibodies. So protein-based biologics are have some their own unique features and drawbacks, right? So the binding site for a protein is much larger than a small molecule, and so this much enhanced surface area means it's just harder to get non-specific interaction with an antibody. You have just more opportunities for the binding interface to go wrong if it's binding to something that you don't want it to. And they tend to have pretty favorable safety profiles. You always have on-target tox issues, but in general, it's made of protein. Your body knows what to do with it, and so you don't have to worry about breakdown products, things like that. But large proteins, you're generally injecting these to administer them to patients, and compared to small molecules, they're pretty fragile in that you need to refrigerate them, and oral administration is pretty challenging. There's not been any successful orally administered antibodies to the clinic yet. So mini proteins, I like to think about as something of a hybrid molecule, where we're still in the large molecule space, but we start to blend some of the properties together of large and small molecules. And we're still, I would say, in the size range that you could consider this a peptide. But what sets mini proteins apart from the really cool cyclic peptides that Yushan just described is that the mini proteins adopt a stable tertiary structure. So they're not often as dynamic of a structure. It's really a rigid three-dimensional structure that these molecules fold into. And so that's much more like a large molecule type binding behavior. And so the binding surface area, it can be quite large with mini proteins. And so we can get really hyperspecific binding. It can be really, really tight. And you can use it to inhibit protein-protein interactions like you maybe would with a large molecule. The structure also is hyperstable. So you start to get some more small molecule properties, and that oral administration starts to become possible. You can boil these things, they don't really care. The ways that you can manufacture them start to become easier using synthetic biology and yeast fermentation. So you can start to approach manufacturing cogs that are closer to small molecules and sort of storage and shipment behaviors that are more closely you would imagine associated with a small molecule. So this is a really fun and unique space to be working in. So we engineer mini proteins, and the process that we tend to do this is 3 steps. And for those coming from the antibody space, and perhaps some folks in peptide screening, this won't be anything super surprising. The main steps where we're doing things a bit differently is in step 1 here is the library construction. So step 1 is we create a library of mini proteins. Step 2 is we screen through that library to find binders. And then step 3 is we optimize them. The library strategies that we employ, there are 2 different ways of doing this. The first one is what we call a naive library, where the mini protein library contains many, many variants. In this case, we're operating at around 1 billion. And normally in those size ranges, you would think about having sort of random diversity. And it's sort of random, except that our strategy is a bit unique in that we carefully curate the randomness. So every random member is one that we explicitly decided we wanted. So our randomness, everything looks like a binding site. So we're able to filter out also a lot of the sequences that are typically promiscuous or problematic. We just don't have them in our library. And so this has the potential to bind lots and lots of different things. Alternatively, we do targeted design as well. So for instances where you have a lot of structural information about the biological system that you're trying to drug, you know exactly where you need to bind. Maybe there's multiple conformations and you want to only bind one of them. We also do targeted design where we can take a specific site that we want and wrap a protein around the binding site. The success rate, this is quite hard, and the success rate is fairly low, so you need to test lots and lots of designs to make this work. So either way, we're building libraries. This library is 10 to the 9 range, and the targeted approach is more in the 10 to the 5 range for diversity. The next step is discovery. So we're using display technologies. So this also means we're sticking, by and large, to the vanilla 20 natural amino acids. We have some ways of incorporating non-canonicals, and it's possible to do that. And there are some instances where you would want to, but usually that's a post ex facto at the end. We're adding a non-canonical, and we're not doing that up front. That greatly limits the ability to use synthetic biology tools if you have to have non-canonical amino acids. And we can do a lot with the natural 20. And so we're using either yeast or phage display to screen through the library and identify our hits. And then the next step, we're doing optimization. So either just kind of standard directed evolution approaches, or we have some unique strategies for saturation mutagenesis where we can test every possible mutation that you can make to this molecule in a single experiment. Okay. So this is somewhat of an obvious concept, but I want to bring this to the front of mind to make the next point here. So when 2 molecules interact, they need to do so with shapes that fit together really perfectly, right? Much like a lock and a key. And in order to take a binding activity that's, maybe very weak and they can weakly associate into something that's really, really very tight binding affinity, you have to be able to change the shape of the mini protein to do this, right? And so this is simply, a 2D representation shape change, but this is a complex 3-dimensional shape change. And there's also chemical complementarity as well, so just charge, hydrophobicity, et cetera, right? So you need to be able to have a lot of precise control over the shape and structure of the mini protein in order to be able to tune its binding activity and to confer novel binding activities. So I explained about why mini proteins are awesome and what they can do and all the advantages. So, why don't we see these all over the place? Why aren't there just a ton of mini protein drugs? And the truth is that the structures are very challenging to engineer. Many of you in the peptide community may be familiar with these venom peptides already. And so this is a natural mini protein. This one actually comes from a squirting cucumber. It's a trypsin inhibitor. And this is a really cool structure here. So I'd like to bring your attention to a couple points. The first one is I've highlighted all of the nonpolar amino acids in this structure. And you can see there isn't really a hydrophobic core. There are a couple hydrophobic residues that are near each other here. I wouldn't necessarily call that a core. Instead, you've got a bunch of disulfide bonds in the interior of this structure. And so this tertiary structure is reliant upon the presence of disulfides in order to get it to fold. The next feature to highlight is that there's little to no secondary structure in this molecule. You've got a lot of loop. And for an antibody, a loop is something of a superpower. And that's because the conformation of the loop changes depending on the amino acid composition of the loop, right? So when you make substitutions in a loop, you change the structure of the loop. And if you're in an antibody and those loops are anchored in a really well-folded domain that is constant, the loop can have all sorts of different shapes, and that can help you bind all sorts of different targets. Well, the mini protein doesn't have this large IG domain to anchor the loops. Instead, at the end of these loops, you've got disulfide bonds. And so when you start changing the shape of this loop, you start shifting the position of this disulfide bond that's required for this molecule to adopt this shape. If the disulfide can no longer form because the loop has torqued it out of its position, then the structure essentially falls apart. So once it goes into this shape, it can be very, very durable, but this shape is also very brittle to mutation. So it's quite challenging to repurpose these. It's only been done a handful of times in the world. Pretty much Jennifer Cochran is one of the only, there's the only folks who's really done it. And even her group has only done it a handful of times. So just because one of the best protein engineers on the planet has been able to do it once or twice, doesn't necessarily mean this is a easy thing that becomes a platform that can solve lots and lots of problems. So this is the problem that we set out. We wanted to make mini proteins a general platform that you could use to create molecules for whatever challenge that needs to be solved. So we did this using de novo protein design. So here's an example of a fully synthetic disulfide-rich mini protein that we designed de novo, and we design this in silico, and then we make it in the lab and test it to see that it folds up as designed. This is an example of a real one here. We've got the PDB code in the bottom corner. And you can see right away that our synthetic mini proteins are quite different. The first is that there is a hydrophobic core. So you can see here the hydrophobic amino acids that are in the interior that direct this protein to adopt a specific shape. The second feature to call your attention to is that we have a lot of secondary structure. And so this ideal secondary structure is really helpful for making the surface of this molecule resistant to amino acid substitutions. So in this helix, for instance, all the amino acids in this helix work together to cause the protein to adopt this shape. And the residues on the backside of this helix that point towards the core also help to keep the whole structure in shape. So what that means is that we've got positions on the surface of this helix that point out into the solvent that essentially, as long as it's not an amino acid that breaks the structure like a proline, you can essentially mutate these positions freely, and it doesn't impact the underlying three-dimensional shape of the mini protein. And so this means that we can create a stable three-dimensional structure where the surface is our playground, and we can make essentially any substitution we like on the surface in order to change the binding activity or to tune the binding affinity. And I'm showing disulfide bonds here and multiple disulfide, but we actually design these things so that they fold up, and this structure can be hyperthermal stable in the absence of disulfide. So we actually don't need disulfide bonds to get this molecule to have a melting temperature north of 80 degrees Celsius. And so we can add disulfides in to go from hyperthermal stable to ludicrously stable. And I'll show some data a little bit later on about some of the just crazy stability that you can get by adding disulfides. Okay, so now I'd like to just give you a bit of idea on how we design these mini protein structures. So we're doing this in silico, and the very first proof that this could be done at all was done using a method we call blueprint builder. And so by this approach, you take a protein structure, say here's our target topology that we want to create, and you can take this complex three-dimensional structure and then break it down into a human readable blueprint. So here, we're counting out the number of beta strands that we want and their arrangement in primary space, as well as how the strands pair with each other. And then we can take this human readable topology map and turn it into a machine readable blueprint for creating a protein of this shape. So here we're defining the pairs of secondary structure. So here we've got strands 1 and strand 3, as you order them in primary space, are pairing with each other. They're anti-parallel, and say we don't want any register shift. And then we define at each position in the structure what we want the Ramachandran angles to be. And so in the beta region, we want these to be in the beta region of the Ramachandran space. You'll see that I use this shorthand a lot, an E for extended, which means essentially beta strand, and an H for a helix, which is an alpha helix. So once we have our blueprint, we can go through the protein data bank and find fragments of amino acids. So we look at all the protein structures and we find stretches of structure that has patterns of phi-psi angles that match our blueprint. We throw out the amino acid information. We don't care about that. All we want is the pattern of phi-psi angles in the backbone. We call these fragments. And then we can take these fragments and stitch them together to create a novel three-dimensional structure. And so while each really small piece of main chain in this case comes from a natural protein, it's really just the pattern of phi-psi angles, and the overall arrangement of these phi-psi angles to create the full three-dimensional structure is completely novel and has never been seen before. So once we get this shape, the next step, if when we're designing in disulfides, is to search for all the positions in this backbone that are able to host a disulfide bond. So we're essentially just doing this through geometric analysis. And then we also have some tricks for spacing them out. So we look at the disulfide entropy. So we try to space the disulfides out. And then finally, we use Rosetta simulations to figure out what the best amino acid identity is at each position. And so we do some obvious things, like we stick polar residues on the surface and non-polar residues in the interior. And we do iterative rounds of Monte Carlo sampling of amino acid rotamers with minimization of the backbone and side chains in order to find an optimized structure and an amino acid sequence that should direct this main chain to adopt this structure. So I showed one example, but we wanted to show that this was a generalizable approach that you could use to design mini proteins of any arbitrary topology. So we picked every permutation we could think of 3 strands and 1 helix. We also did a couple versions that were comprised only of helices or only of strands. And then we removed one of the strands to do just 2. You can see this topology, we were able to get a parallel beta sheet as well as an anti-parallel. So we made hundreds of designs in the lab. Some worked, some didn't, and we picked one representative structure, one representative design for each topology to move forward for a really detailed characterization. One of the nice things with these is because they're full of so much canonical secondary structure, we can use circular dichirism spectroscopy, CT spec, in order to assess the secondary structural content. So I'm not sure everyone here is familiar with CD-SPEC, so I don't want to belabor it too much. I'll just maybe focus all of your attention to this corner of the screen here. So here, this is a peak that's really canonical mixed alpha beta topology. We have a broad negative peak between 208 and 22 nanometers. And so this indicates we have both alpha helix and beta sheet. When we heat this sample in the spectrometer up to 95 degrees Celsius, you can see the secondary structure elements flex just a little bit, but they doesn't denature. And then when we cool this protein back down, it snaps right back into shape. And so this is indicative of a hyperthermal stable structure. And then when we blast it with a really strong reductant to break all the disulfide bonds in this, we convert into a random coil signature. And so this particular structure is reliant upon the disulfide bonds in order to maintain its thermal stability. Other ones are not. This is an example of a structure that when you break the disulfides, doesn't seem to change the structure too much. And then we've got all sorts of things in between. We repeated this with chemical denaturants. And so here we titrated in guanidinium hydrochloride and all the way up to saturating concentration, 6 molar. And so you can see here, call your attention to the same structure. Even at saturating concentrations of guanidinium hydrochloride, this structure remains intact, and we're seeing very little to no change in the secondary structure content. So this means that these proteins can be insanely stable and resist not only extreme temperatures, but also chemical denaturants. Their tertiary structure remains unchanged, or secondary structure remains unchanged, and presumably their tertiary structure as well. Okay. So we wanted to really test and see if this was working, and so we determined structures for 7 out of those 9. And I'm showing here, in each case, the computationally designed model in rainbow color, and the experimentally determined models are in gray. This design here is a crystal structure, crystallized with 3 copies in the asymmetric unit, and so they're all shown aligned here of this C alpha trace. And the other 6 models are all NMR structures, and so you can see all the models of the NMR ensemble are aligned against the main chain here. In each case, our C alpha RMSD is around an angstrom or less from both inside the NMR ensemble and the model to the NMR ensemble. And so essentially what this means is our ability to design these structures is accurate to within our ability to measure it. And on the right side here, I'm taking the lowest energy experimentally determined model and aligning it to the computationally designed model. And this may be a little bit hard to see, but in many cases, we're even getting I'm showing the nonpolar residues in the core as showing their side chains, and in most cases here, we're even getting the core monomers correct. So all this is to say, we can really design these mini proteins. We can design them in a lot of different shapes, and we have the ability to do so with a sort of extreme accuracy. So what's next? What are we going? How are we designing these mini proteins to make useful drugs out of them? So one of the big challenges that we face is that blueprints are all nice and good if you know exactly what structure your mini protein needs to be in order to solve whatever challenge that you're trying to solve. But that's usually not the case. It's quite uncommon to know exactly I need this precise blueprint in order to solve my problem. So we set about creating a new method for designing mini protein structures that doesn't rely on blueprints and allows the algorithm to figure out what the structure should be. So this is loosely based on the sewing algorithm, for those of you who are familiar with Rosetta. That originally came out of Brian Coleman's lab, although we've really kind of built this from the ground up. I'm showing it here that using fragments, although we've actually got this working now without any fragments at all. But the basic concept is that you take structural elements of proteins, again, just patterns of phi psi angles, and you can stitch them together to create entirely novel topologies. And we can have a large library of these fragments. We can do lots of sampling, and we can create a wide variety of different protein structures. And without having to know what the blueprint is a priori, we can create novel structures to satisfy functional criteria. So in the case of creating a library, our functional criteria might just be being different from all of the other members because we want to maximize the geometric diversity in our library. Or for a targeted binding approach, you might need to be able to hold specific amino acid side chains in a geometric orientation that's compatible with target binding in the site. And so we want to wrap a protein around those amino acid side chains, and we don't particularly care what that three-dimensional topology looks like. It just needs to be able to hold those side chains. And so this approach lets us do that. And so I'm sorry I'm not talking a whole lot about AI in this AI symposium here, but this is one of the breakthroughs that we've made at AI Proteins is an algorithm that can look at this protein structure and tell if it looks like a protein or not. So this has been a challenge that the de novo protein design field has struggled with since the very inception, and is by and large relied on human intuition in order to do this. So you can see that there's, let's look at this shape here and this shape here. The human eye, when you look at this, you can tell this structure is garbage. It doesn't matter how good we are at putting amino acid side chains on this. There is no combination of amino acids that you can stick on this backbone and have it form this shape in a stable way. It's just too open. You're not going to be able to pack nonpolars to form a core. It's a bad topology. And I can tell that before we even put amino acids on it. So just from the backbone alone. But it's been really, really hard to teach a computer to be able to do this. And so this is what we figured out. And so you can see this is a per residue score. This structure, which is much more compact, has the potential to have really good amino acids designed into it to form this structure stably. We can now identify those in a pre-designed stage where the geometric sampling is fast and cheap. And if we like, we can perform iterative rounds, change the size of these to be small or as large as we like, and grow this structure as small or as large as we need for whatever problem we're trying to solve. So this is all nice and good, and wanted to do some proof of concept to show that this approach works. It's not so crazy. And one of the things I wanted to do is go back and try to fix something from the previous work that was just kind of nagging at me. So, I'm showing this here just with helical fragments, although we have this working with also beta sheets as well. But in the initial work that I showed you before, I kind of glossed over these all helical ones. I want to focus on the helical designs, as this is wasn't it very good when we did it before. And so you can see when we heated these up, they actually flexed quite a lot. And when we added chemical denatures to these really readily denatured essentially. And when we got to the saturating concentrations of guanidinium hydrochloride, they were basically completely denatured. The secondary structure disappeared. So why were these helical ones not very stable, and can we do better? So we took our new design algorithm and we added some layers on top. And so this is one way that we can use to assess the structure after we have a sequence on it, is to look at how well the core is interdigitating. So if you think about a helix packing on another helix, it needs to do this with like a knob and hole kind of arrangement in order to be really, really stable. And so you can think about this as like two gears packing against each other, right? We want to have this knob and a hole. And so we wrote an algorithm that looks at each helix and it says like how well is this packing against every other helix in the structure. So we call these elements, and then we look at essentially the shape complementarity of the core residues on this interacting with the core residues on this. And this will let us know if we've got a well interdigitated structure where all the structural elements are packing against each other. So we created a library of 1,000 of these structures in silico, and then we tried to put disulfide bonds in them. And we wanted to have a disulfide bond at each terminus. So one disulfide bond near the N terminus and another disulfide bond near the C terminus. And it was okay if it was the same disulfide bond. We just really wanted to make sure the termini were locked down. The algorithm that we use to do this, it's called disulfidize. It's called the disulfidizer. Yeah, kind of cheeky, but we wanted to just, it's adding in a bunch of disulfides, so that felt appropriate. And so the term here is, can we successfully find all the disulfides in our structure that we want to? We say that structure then disulfidizable. Bit of a tongue twister. So we designed 1,000 mini protein structures that contain 3 helices and 1,000 mini protein structures that contain 4 helices. And then we looked out of those structures, how many of these were packable, so able to satisfy the filter that I just described here about interdigitation of the helices against each other, as well as could be disulfidized and have disulfides locking each terminus. And we were a bit surprised to find that the 3 helix bundles, we couldn't attach any disulfides at all in here. And the 4 helix bundles was difficult. We got a couple of them, but none of them also had nice cores. So this was a bit odd. So we decided to dive into this a little bit further, and we looked at the entire PDB, and we looked at all the disulfide bonds in the PDB. And what we did is well, first we curated a non-redundant PDB because there's lots of structures of the same protein over and over again, bound with different ligands or maybe with point mutants. And so we didn't want to have the results too skewed by just overrepresentation of some structures. We're still getting that to some degree, but we wanted to minimize that as much as possible. So we curated a non-redundant PDB and then looked at all the disulfides, categorized the secondary structure that each residue in the disulfide pair was in, and then here's what we have here. So in red, you can see the most common disulfide in the PDB is between beta strands, and the second most common is between loop residues. The rarest disulfide in the PDB is between helices. That was interesting. We then took our non-redundant PDB and we ran the disulfidizer on it. And so then we found all positions in the PDB that are geometrically compatible, all pairs of residues in the PDB that are geometrically compatible with having a disulfide bond engineered into that site. And we saw essentially a very similar pattern. And again, disulfides between helices were the rarest of all the disulfides that are geometrically accessible in the PDB. So as we started to think about this a little bit more, it just seems like nature doesn't like to put disulfides between helices, so maybe we shouldn't try to force it. So because we had the ability to extend and control these shapes, we thought, well, maybe we don't need to force the disulfides in here. What it seems to be is that in order to get good helical packing, maybe the geometry of the disulfide formation is just not compatible with the geometry you need to get good knob and hole packing. Makes sense. So what about if we cheat and we just extend little bits of loop fragment off the ends? So in this case, we can just grow some structure, look for disulfide bonds, check to make sure this is good, and then we can add disulfides that essentially staple the termini, which can be just as essentially accomplishing what we were trying to do already, but avoiding the problem of making disulfides between helices, because a disulfide between a helix and a loop is far more common. So when we took the same set of 1,000 3 helix structures and the same set of 1,000 4 helix structures, and we ran our loop extension algorithm and then looked for disulfides, now we got a lot more disulfides in these structures, and we started to get significant and substantial overlap between the structures that have good packing and the structures that we can now disulfidize. Sorry, this is a typo. This should be a 1 here. So this was really exciting, and so now we can overcome the mutual exclusivity between good disulfide geometry and good core packing and helix interdigitation. So this really works. Here's a design, just the main chain topology of one of our designs. You can see we've got 2 disulfide bonds, 1 at each end. And here's CD spectra at varying temperatures. You can see we're much, much more stable. We still flex a bit at high temperature, but this snaps right back. And just to kind of show you some of the extreme nature of the stability that you can get by incorporating disulfide bonds, we ran an experiment where we incubated our mini protein with synthetic gastric fluid. And so here we've got this disulfide-rich mini protein, and here we have a couple of disulfide-free version mini proteins. I'm not showing their structures. They're similar 3-helix bundles, but they don't have these disulfide bond extensions on the termini. All of them have been validated by CD-spec, and all of them are hyperthermal stable. So in this case, we incubated these structures either with just phosphate buffered saline at pH 7.4, or with synthetic gastric juice at pH 1.5. We incubated these for 2 hours at 37 degrees, so human body temperature. And you can see the disulfide-free ones are largely hydrolyzed in these conditions. And when we desalt the protein using zip tips and then run them on an STS page, the disulfide-free variants have essentially been hydrolyzed by the acid, and you don't see them anymore. This one's still there a little bit. The structures are quite stable, but it's certainly been diminished quite a lot. And our disulfide-rich mini protein, on the other hand, still there, highly resistant to hydrolysis by acid. So this is very exciting, and we're using these molecules as engineering platforms to create high affinity binders to things. And part of that I mentioned is yeast display. And just to show you that the disulfide-rich loops don't interfere with our ability to perform yeast display experiments with these. This is just taking the number of cells, displaying the mini protein on the cell surface. We've got a population of cells. We run them, we label them for display on the cell surface, and then push this population of cells through a flow cytometer. With yeast, you normally don't get it, about 50% of the cells at best will display a protein at a given time, simply because cell surface display is tied to the yeast cell cycle. And so our disulfide-free ones, we're getting really great display, and our disulfide-rich mini protein, we're also getting really good display. Here you can see just the fluorescence intensity of the non-displaying versus the displaying populations. So we're getting really strong display with these, and the presence of the disulfide isn't changing the strength of the display. So we can make really hyperstable proteins and get them to work really well in display technologies. So that's the story that I have to share with you today. Hopefully I've convinced you that mini proteins are an exciting opportunity to create new therapeutics, and that we can combine some of the really cool features of small molecules and large molecules and unify them into one new modality. We can design these structures de novo, and our ability to design them de novo enables us to functionalize them, and that the next generation of de novo design approaches can allow us to have improved properties and allow us to design them to have more requirements for the structure. So thanks so much for your attention today. Thanks for the team, my lab, that decided to take the plunge and leave academia with me and come to form this company. And yeah, thanks all of you for your kind attention today. I'm excited to take questions.
Thank you, Chris, for the presentation. That was really interesting to hear about the mini proteins. Let's see, do we have any questions in the chat? Okay, so Charlotte has a question. After seeing the previous talk, I wonder how you take into, is this a question for Chris or for you, Shan? I think this is a question for you, Shan, sorry. Okay, so here's a question for you, Chris, from Christopher. You have the capability to design very diverse and target-specific alpha helical mini proteins, but you showed that some helical bundle topologies have better properties than others. When you screen a new target, do you screen many alpha helical bundle topologies or just a small subset of better de-risked ones that you have previously tested?
Yeah, more is better, basically. So the more diversity you can put into a library, the better, always, yeah.
Yeah, I have a question, but I think you might be biased. So Rosetta or AlphaFold?
Oh, well, so let's see. That's also a great question. It depends on what you're trying to do. So AlphaFold is a structure prediction tool, not a protein design tool. There are many people that are hacking it to run it backwards and use it for design. but that's still very much in the academic realm, and it awaits validation that it really works. So AlphaFold also, it's machine learning, right? So ultimately it's memorizing protein structure and telling you back protein structures that are all look like natural things. So in order to get really crazy alien shapes, you won't be able to train on natural structures to do so. So there are some strengths and some weaknesses, but at the moment I would say Rosetta, for structure prediction, AlphaFold all the way. For structure design, right now I would say Rosetta is the stronger tool, but I'm sure that will flip and there will be perhaps not AlphaFold, but other things that will supersede it.
Yeah, no, I agree. I think that's a very good answer. Yeah, for structure prediction, I think we may see slightly better performance, but yeah, definitely for design, Rosetta. I was curious, I don't see any other questions, so I'll ask another one. I was curious if you can allude at all, and it's okay if you cannot, to any of the training data sets that you guys use to make the stability models.
Right. Yeah. So a lot of it is also trying to take a first principles understanding and combine that with some ML heuristics. So yeah, it's kind of hard to go into more detail. Sorry.
Yeah, no worries. No, I wasn't sure if you'll be able to talk about it. Okay, so we still have a lot of people on the call today, so I am looking for more questions because I'm out of questions. So please ask questions. Maybe Yushan has a question.
I guess no, I think I was just looking at Charlotte's question. I think maybe she is wondering if the mini protein are they really super rigid, or they are still some conformational flexibility. And I think which is probably related to what I might be thinking, that is for the loop, is it also very well organized, or it's flexible, and then would that somehow play in our favor?
Right. Yeah, that's a great question. So we try to make these structures as rigid as possible. So there's not a lot of motion going on. We try to freeze out as much as we can. Also, the loops are tight canonical turns. So there's very little room for dynamism in the loops. For a de novo designer, loops are the hardest part. They're very scary, and so we try to keep them as short and as simple as we possibly can, because that tends to be the make or break for most designs.
I see. And then I saw that you extended the helix a little bit, and added disulfide bonds. Somehow reminds me of like a helix cap or like helix nucleation kind of theory and then also I guess also about like stability. Seems like the ends are the weaknesses, and then tying the end to some anchor seems like a very smart and efficient move to stabilize helix bundle.
Yep. We also do try to include the canonical and terminal helix capping residues. So we are requiring the loop preceding a helix to be serine, threonine, aspartate, or asparagine in order to be able to provide those those capping interactions with the backbone.
Right. Is there any interest in using some linkers that other than disulfide bonds, so it's less sensitive to oxidation?
Yeah, we've explored diselenide bonds and such. And that's sort of easy to just kind of swap out. Yeah, other linkers can be tricky. For us, we think about like the vanilla 20 amino acids is where we like to stay, simply because we can perform directed evolution and we can do yeast display and test billions of variants, and that allows us to operate in scales that we just can't do with if you have to synthesize them on a peptide synthesizer. So I'm I know woefully boring when it comes to the chemical matter that we're using in these peptides, but yeah, we're trying to stay genetically encodable.
Yeah. So there's a couple more questions. One's from Ved. Could binding affinity be predicted with mini-protein when docked with a known protein target?
Yeah, that's a great question. We've tried, so we do predict the structures of our mini-proteins with AlphaFold. It's unclear how much signal there actually is of like true positive versus true negative rate. It seems to be very weakly correlated just with the predicting the structure of itself with actual success in the laboratory. We're doing it for now more fear of just, what if we don't do it, even though we don't think it actually adds much signal. And then predicting the structures, we've also tried taking our binders and doing a complex structure prediction with the target. And yeah, it doesn't do a very good job. Some of it is that AlphaFold requires coevolutionary information really to make its predictions quite well. So you generally start with a multiple sequence alignment when you're doing AlphaFold. And so you need to have a lot of evolutionary history in order to make an accurate prediction. And because these are de novo, there is no evolutionary history. So the accuracy of the predictions is basically.
Yeah, no, that's super interesting. I mean, at the end of the day, I think, this is an AI talk. Training data is everything for AI, right? Yushan can probably attest to that. If there's not good diversity and coverage in the training data, you will never get really accurate predictions. So there's one more question from Chris. So mini-protein ultra stability seems like a great thing for in vitro experiments, but is it possible being too stable in the pharmacokinetic liability in vivo? Meaning many proteins will not be cleared or they will aggregate.
Yeah, also a great question. So it depends on what you're trying to do with them, I would say, but by and large, mini proteins on their own will very rapidly clear through the kidneys, right? It's below the size range where glomerular filtration retains large molecules. So these will be rapidly excreted through the urine. Their residence time on target, if you have the binding affinity really tight, can be very long. So we can be in like the week-long half-life range bound to target. But ultimately, the mini protein will fall off and clear through the kidneys. And at the end of the day, it's still made of protein. While it's certainly much more durable, when it's out in the environment, eventually it's going to break down because it's still just amino acids.
I see we have one person who raised their hands. I'm going to let them. You should be able to unmute. You can unmute yourself if you want to ask a question.
Nothing. I'll ask my last question then, maybe. So you showed the gastric stability of some of these mini proteins. There's a lot of diseases, of the small intestine that could be really great targets for your mini proteins. I was wondering if you're also going in that route where you're trying to basically go after specific targets for the GI.
Yep. Yeah, you'll probably not be at all surprised to hear that, yeah, inflammatory bowel disease is an area that we're actively pursuing. But yeah, excellent point.
I think that's about it. We're came to the end of our time. Thank you for coming. I don't know if Yvonne or Becky want to say any closing remarks.
Sure. Yeah. Thank you, everybody who attended. Thank you to our speakers. Really great virtual seminar today. And just want to remind everyone that we do have, I think, one more virtual seminar in August. Becky, do you have those dates or anyone?
I believe it's August 2nd. I want to say it's 4 p.m. Eastern time, but maybe Becky can remind me.
Thanks, Laura.
Yeah, I'm pretty sure that that's right.
And
So-
we will promote it through email and on the website, and you should,
Yeah.
everybody should sign up.
Yes, so look for that, our next virtual seminar.
Yeah, Yvonne, it's August 2nd at 4 o'clock Eastern time, and that's the cyclic peptides related to the PCSK9 story.
That's right. So we look forward to seeing everybody then as well, and hopefully you can join us in person November 7th through 10th in Boulder, Colorado.
And the registration's open for that on the website.
So thanks again to all the attendees and to the speakers. Really exciting work. Thanks so much.
Thanks, everyone. Good questions and good talks.
Thanks.
Bye.
Thank you.
Take care.
you.
Bye.
Bye, Lauren.
Bye.